Hello everyone, and thank you for joining for this presentation. My name is Orestis Stavrakidis-Zachou, and I work at HCMR, the Hellenic Centre for Marine Research, on things related to fish physiology, climate research, and bioenergetic modeling. What I will talk about is some trials we have conducted at HCMR in an attempt to understand how fish, and specifically the European sea bass, are affected when they are transferred from marine cages to tanks. First of all, why should we care about this, about the effect of transferring on fish? Well, we do because we know it is a very stressful procedure for them. Most fish will be transferred at least once in their lifetime, and that is when they are moved from hatcheries and pre-growing facilities to marine cages for rearing. But also sometimes for experimental purposes, there may be a need to move fish from marine cages back to land to the labs where the actual experiments take place. And we know this is very stressful for the fish. Not only handling is a physical stressful process, but also changes in the environmental conditions such as temperature, pH, salinity, and so on, that are rarely identical between the sites, may cause additional stress to the fish because now they need to regulate their physiology to cope with the new conditions. And this can be particularly important when we're transferring fish from marine cages to tanks. It is also established that if we want to have quality results from any fish trial, we must use fish that are unstressed and fully acclimated to their environment. Therefore, two questions arise. How does this whole transferring procedure affect them? And how long does acclimation last? In order to answer these questions, we've conducted two trials at HCMR regarding the European sea bass. Specifically, we wanted to see whether there was a size dependency on the acclimation period. And for this reason, we did a trial using small European sea bass at about 100 grams, and also another trial using slightly bigger fish at 300 grams. For those trials, we took a number of fish from the pilot scale cage farm that the Institute has and transferred them all the way to our land-based experimental facilities. We did these using specialized tanks, and the duration of the transfer was approximately 3 hours. After that, we placed them in tanks and monitored them for a period of 8 weeks. Now, what we monitored was husbandry variables such as feeding, which relates to appetite and mortality, also changes in physiology, which we did by collecting and analyzing blood samples, and also we monitored behavior. For this, we used overhead cameras to record the swimming activity of the fish, and then used a method to assess things such as the swimming speed and the preferred swimming level in the tank, and also the shoaling behavior. So what did we learn from these trials? First of all, for the small European sea bass, we saw quite immediately that they seem to adapt quite easy, and we recorded almost no mortalities. This was also verified by physiological findings. Here you can see how the cortisol levels, which are an indication of stress, changed over the course of the 8 weeks. Compared to the control measurement right at the beginning, before the transfer of the fish, you see that there is a slight elevation right after we place the fish in the tanks. However, after the first week, these values return back to normal. So we could say that physiologically speaking, adaptation happens quite fast within a few days. Regarding behavior, again, it appears that it took only about a week for the fish to return to behavioral normality. In the graph, you can see how the swimming variable changed over the course of the trial. If we notice at the red dots, for example, which indicate the swimming speed, at the beginning, the fish were very sluggish and did not have a high swimming speed, but within the first 7 to 10 days, they resumed normal behavior. And the same goes for their shoaling behavior with the green triangles. At the beginning, they aggregated close to each other and the fish displayed this high shoaling behavior. But after the first week, they started moving randomly in the tanks. As for husbandry, and here I'm referring to how fish were feeding and how their overall appetite was, we see that acclimation required a bit longer. For the first week, the fish were not feeding much, and it required a second and a third week for them to return to normality. Therefore, overall, we can see that physiologically and behaviorally, small European sea bass adapt quite fast, and they need a bit longer in terms of husbandry. As for the bigger European sea bass, those showed more difficulty adapting, and we did record significant mortalities for the whole first week after we transferred them. However, if we look at physiology and behavior, we observe the exact same patterns that we saw for the young European sea bass, so the fish adapt quite fast, and it seems to be something that the species does in general. However, in terms of husbandry, appetite was almost non-existent in the first week, and the fish fed very little for the whole second and third week. So overall, to fully acclimate, larger European sea bass needed over a month. So is this information that we produced useful, and to who? Well, for researchers, this information could be used to design better experiments, which in turn can lead to more reliable, more robust results by ensuring that the fish we're using for our experiments are in the right condition for the experimental purposes that we want them to be. As for the fish farmers, they may also benefit from this information by timing their transfer operations better, by minimizing stress and mortalities, and also by reducing feed losses. And this goes again to the appetite that was discussing before. As we saw in the case of the large European sea bass, changes because of transferring procedures can be quite substantial. Finally, I would like to end with the potential economic impacts of the results I presented. For researchers, this knowledge may be useful for optimizing experimental resources, which saves money in the process. While for fish farmers, this information also may be used for improving husbandry efficiency and managing mortalities and feed losses, which in turn could lead to lower production costs and maybe even higher profits for the farm. I should note, however, that we should be cautious when extrapolating these results, because this trial was done for European sea bass, and therefore the results may be very species specific. Therefore, similar trials need to be done for other species, and there may be a need to also do tests to see the opposite effect, which is the effect of transferring from tanks to cages. And with that, I'd like to thank you for your attention and for showing interest in our work. My contact details are on the screen, so feel free to contact me should there be any questions. Thank you very much.